Hello everyone. Now we are going to move on to the third KIMS in the biotechnology principle and then process. So still now in our first KIMS we learned about what is in a biotechnology, and then in the second KIMS we have learned about restriction enzyme tools which is used in a recombinant DNA technology. So in that first one, restriction enzyme. So now we are going to move on to the second tool, that is called as a cloning vector. So this is a one of the cloning vector which is called as a PBR322. So this is a artificially made cloning vector. So before going to our content part, so first we will be clear with the prerequisite. So what is a word? What is a recombinant? The recombinant is nothing but we are going to recombine a particular genome which is called as a recombinant. So this is a one plasmid. So we are going to cut a one region and then we are going to introduce a gene and then this become a recombinant DNA. So this is a recombinant. When it is going to be non-recombinant, which doesn't has our gene or a gene of interest. So that is a non-recombinant. When it is going to be a recombinant, they has a gene of interest. The next one is antibiotic resistance. What is that antibiotic resistance? So usually antibiotics which is used to kill the microbes. Some microbes they have this antibiotic resistance. So let us take an example of this is a one antibiotic capsule. So what happens means we are going to introduce into a one culture medium. In that culture medium that bacteria will be growing. So when we have put this antibiotics, they are going to easily kill the bacteria if they don't have any restriction power. But if they has a restriction power, they cannot be easily killed. So that is called as a antibiotic resistant property. Some bacteria they exhibit this antibiotic resistant property. So we are going to see this antibiotic resistant property which is going to be play one of the role in a cloning vector. So antibiotic resistant means which is get resistant to the antibiotics. The next one is a cloning. What is a cloning? Production of a exact identical copies. That is called as a clone or a cloning process. So coming to the objectives, so we are going to study about the cloning vectors. So this is a making of a cloning vectors. So we are going to cut a plasmid and then they are going to introduce a gene of interest. So we are going to seal it and then they become as a complete recombinant DNA. So now we are going to move on to the second tool in a recombinant DNA technology, that is a cloning vector. So what is a cloning vector? So why it is called as a cloning vector means because they has a ability to replicate within a bacterial cell independent of the control of chromosomal DNA. So this plasmid or either a bacteriophage or a plasmid, some of the vectors also in a biotechnology field. So two examples, one is a plasmid, another one is a bacteriophage. So these two also called as a cloning vectors because they can able to replicate within the bacterial cell independent of the control of the chromosomal DNA. So they are not controlled by this chromosomal DNA. So they can be easily replicate without any destruction. So that is why it is called as a cloning. Vector means it which is used to carry the gene from a one bacteria to the another bacteria. So that is why it is called as a cloning vector. So one example is a plasmid, another one is a bacteriophage. So bacteriophage, what is a bacteriophage? The virus which is used to infect the bacteria which is called as a bacteriophage. So we are using this virus to infect the bacteria. So this is also called as a one of the vectors. So bacteriophages has high numbers per cell. So they have very high copy number of their genome within a bacterial cell. So during that infection, when they are going to infect a bacterial cell, they can able to produce a more number of copies and then they can be make their progeny to be more in a number. So this making as a one property which is used as a cloning vector. So some plasmids have only one or two copies per cell, whereas others may have a 15 to 100 copies per cell, even more than this. So this criteria makes this plasmid to become as a cloning vectors. So they too has a cloning property. They too has a vector property. That is, they can able to easily carry the information from a one bacteria into another bacteria, or one organism into another organism. So that is why these two are an example of cloning vectors. So vectors are used at present, are engineered in such a way that they help easy linking of a foreign DNA and then selection of a recombinant from a non-recombinant. So another important function of a vector is they can be easily selected whether it is a recombinant or non-recombinant. So they can able to easily identify whether the foreign gene has been incorporated or not. So this is how the donor DNA, this is a vector. So we are going to digest with the restriction enzyme. So they are going to produce a gap. So this gap can be sealed by a DNA ligase. So this can be easily introduced into a host cell. So inside the host cell, they are going to replicate more in a number. So this is a one example of a E. coli. So when it is plasmid which is get introduced into the E. coli, they can able to produce more number of a progeny. So this plasmid can also able to use to check whether the recombinant has been taken or not. So let us see how this cloning vectors going to be. So this is one of the cloning vector. So the plasmid, which is also one of the example of a cloning vector. One artificially made cloning vector is that PBR322. So what is that PBR322 means? P stands for a plasmid and then BR stands for a scientist. So the 322 means 322nd time they made that cloning vector. So that's why it is called as a PBR322. So basically a cloning vector should has some important parts which is called as a origin of replication in a short form which is called as a ORI. Another one is an antibiotic resistant gene. Another one is a selectable marker. Another one is a restriction site or a cloning site. So now we are going to study about what is this parts going to be work and then what function they has. So now we are going to see each parts and their function. So the first one will be a origin of replication. So in this origin of replication, the replication is going to starts from here. So that is why it is called as a origin of replication. So any piece of a DNA which is linked to in this sequence, which is going to replicate within the host cell. So if a foreign DNA is going to be inserted here, they are going to start the replication process continuously. So this origin of replication, they are going to control the copies number of a linked DNA. So this origin of replication only they are going to control the how much amount of a plasmid or how much amount of a cloning vector should be inside a bacterial cell, because this origin of replication has a property to stop and then start the replication process. The next one is a selectable marker. So what is a selectable marker? So which is used to identify and then eliminate a non-transformants and then selectively permit the growth of a transformants. So what is a transformer and the non-transformants is? Transformants means which can able to transform from their original way into another way, that is a transformants. So the other name for a transformants is a recombinant. So the recombinant means in the recombinant we are going to introduce the plasmid which has a foreign DNA which is called as a recombinant. So likewise, if it is going to be a transformants means one bacteria or a plasmid which can able to transform their original property into another property which is called as a transformants, and then which does not able to transform their property which is called as a non-transformants. So the genes encoding resistance to antibiotics such as antibiotic resistant gene, so they are called as antibiotic resistant genes. So these genes can have a resistance to the antibiotics such as ampicillin, chloroamphenicol, tetracycline or a kanamycin. So these are some of the antibiotics. So if the cloning vector, whether they has this antibiotic resistant gene means they can be grown in a antibiotic medium. So when these cloning vectors, when introduced into an E. coli cells, they got that resistant power. So if it is going to check whether the recombinant has been taken or not, will be checked by this antibiotic resistant gene markers, that is the selectable markers. So these are some of the suitable markers for a E. coli: ampicillin, chloramphenicol, tetracycline, and then kanamycin. So normally the E. coli does not carry any resistant gene. So if they has the plasmid which has the resistant antibiotic resistant means, then only they can able to grow on a selective antibiotic medium. So by this we can able to check for a transformants and then non-transformants. The next one is the cloning site or the recognition site. So what is this recognition site means? More than a one recognition site within the vector will generate several fragments which will complicate the gene cloning. So this recognition site means the site which is recognized by the restriction enzyme. So if there has a more recognition site means which cannot able to cut the plasmid. Let us take here is a eco RV. Let us here also one eco RV means which is difficult to cut whether which region. So that is why they are having any one recognition site for a one restriction enzyme. So the ligation of an alien DNA is carried out at a restriction site which is present in one of the two antibiotic resistant gene. So always this restriction site will be more in a antibiotic resistant gene. So in a selectable marker, this antibiotic resistant gene will be present inside. So inside this antibiotic resistant gene, the recognition sites will be seen. Let us take a one example. So we are going to ligate a foreign DNA at the BAMH1 site. So we are going to use the BAMH1 restriction enzyme to cut this region, and then we are going to introduce a foreign DNA here. So what happens means they are going to lose a tetracycline resistant. Why? Because we are insert a foreign DNA in the tetracycline region. So in a cloning vector, the two antibiotic resistant gene is the ampicillin resistant gene, another one is the tetracycline resistant gene. So ampicillin and then tetracycline are antibiotics. So if they has this resistant gene, they can able to grow in that medium. So when we are going to introduce a foreign DNA in a tetracycline resistant gene area means they are going to lose a tetracycline resistant power. So when we are going plate this cloning vector bacteria, this cloning vector which is inserted into a bacteria. So when we are going to plate this means plated in an ampicillin resistant means they can able to grow because this ampicillin resistant going to work because we are not introducing any foreign DNA here. But when we transfer this same colony into the tetracycline means the recombinants cannot grow in the medium because our gene of interest will be here. So this normal tetracycline resistant power going to be lost. So they cannot able to grow in a tetracycline medium. So that way we can able to select the recombinant and then non-recombinant. So a non-recombinant will be grown on the medium containing both the antibiotics because they the non-recombinants they don't have any foreign DNA or a foreign gene, so they can able to grow in both the medium. But the recombinant will be grown only in the ampicillin, not in a tetracycline. So this is a one example. Let us take an example of ampicillin. So when we are introducing a foreign gene in ampicillin resistant gene means they cannot able to grow in a ampicillin medium and the normal non-recombinant can be grown in a tetracycline medium. So this is where we can able to select a recombinant from a non-recombinant by the help of an antibiotic resistant gene. So that is why this antibiotic resistant gene which is called as a selectable marker. So one antibiotic resistant gene helps in selecting the transformants when another one is get inactivated. So when we are introducing here, it's becoming inactivated. So with the help of this ampicillin, we can able to find out the recombinant or a transformants. So that is why they stated that one antibiotic resistant gene helps in selecting the transformants when another one is get inactivated. Then another alternative selectable marker has been developed, means which is used to segregate the recombinant and the non-recombinant based upon their colors they can able to produce. So the chromogenic substrate means colored products. So let us take a recombinant DNA which is inserted into a cloning vector in a antibiotic resistant gene area. So in that, if a region which is going to produce an enzyme that is called as a alpha galactosidase or a beta galactosidase enzyme. So if the foreign DNA which is going to inserted in the area where they can able to produce a galactosidase enzyme, means they are going to be inactivated, because we are going to inactivate the enzyme because we are going to introduce a foreign DNA in that enzyme region. So that process is called as a insertional inactivation, because when we are going to insert a foreign DNA, we are going to inactivate the enzyme production. So that is why it is called as a insertional inactivation. So if we grow the bacteria in a medium, means if the foreign DNA which is get incorporated into the enzyme production area, means they cannot able to produce the color. If they don't have that foreign DNA, means they can able to produce a blue color. So the plasmid in the bacteria does not have any insert. If they don't have any color, they are producing only white color, means their plasmid has a recombinant colonies. So it is called as a recombinant colonies, because in that bacteria that plasmid going to have our gene of interest. So this is going to be called as a non-recombinant, because if it is going to produce a blue color, means they are going to be a non-recombinant. So if it is going to be a recombinant, means they are going to produce a white color. So if a foreign DNA which is going to inserted in a galactosidase enzyme production, means they are going to produce a white color. If it is not going to be introduced in which is not taken up in the bacteria, means they are going to produce a blue color. The next one is a vectors for a cloning genes in a plants and then animals. So still now we learned about the microbes which is used to carry as a vectors. So for a bacteria we can use a plasmid and then bacteriophage. When it comes to the higher animals, that is the plants and then animal, means they need a separate vectors for us. So one vector for a plant and then one vector for animal we are going to see. So transferring gene into a plants and then animals from a bacteria and then viruses. So we are going to transfer genes from bacteria to plants, means we can how to deliver the genes to transform a eukaryotic cell and then force them to do what the bacteria or viruses want. So we are going to use a one plant vector is called as a Agrobacterium tumefaciens. So this is one of the bacteria which going to be the pathogen of a several dicot plants. So this Agrobacterium tumefaciens can able to cause a tumors in the plant. So they can able to deliver a piece of DNA which is known as a tDNA to transform a normal plant cell into a tumor cell. So if this Agrobacterium tumefaciens which is going to present inside the plant cell, means they are going to produce a tDNA. So this tDNA can able to produce a tumors in a plant cell. So what they did means this tDNA direct these tumor cells to produce the chemicals required by the pathogen. So what are the chemicals needed by this pathogen which is going to be produced by this tDNA once they produce the tumor cells. The next one is a retrovirus. So this retrovirus in animals, which is ability to transform a normal cell into a cancerous cell. So they has a art of delivering genes by a pathogen. So they are going to has a delivering property of a delivering gene property. So that is why they are used to transform these tools of a pathogens into a useful vectors for delivering genes of interest to the humans. So the scientists, they use that Agrobacterium tumefaciens DNA, that is tDNA, and then retroviruses to deliver the gene of interest to the plants and then animals. So this is how the tDNA, which is seen in Agrobacterium tumefaciens, works. So let us take, this is a plant which is get affected by the Agrobacterium tumefaciens. So when we see it inside the cell, means this Agrobacterium tumefaciens, they are going to release a tDNA. So once this tDNA they are going to produce, means they are going to cause an infection, and then they are going to produce a tumors in the plants. So what the scientists did, means they make this tDNA to act as a vector. So they reduce the infection property, that is they are going to stop the tumor producing property, and instead of that they are going to insert a foreign DNA. So once they insert the foreign DNA, they become as a Ti plasmid. So they are going to act as a plasmid in a Agrobacterium tumefaciens. So once they produce this Ti plasmid, they are going to be easily delivered to the plants, because they has a infecting property, delivering property, not a infecting property now, because that infecting property gene has been altered by our foreign gene. So they has the delivering property, and then they can able to deliver the gene of interest into the another plant cell. Likewise, retrovirus also, we are going to change the infecting property, and then we are going to using this delivering property only to deliver the gene of interest from a bacteria to the humans. So this tumor inducing plasmid of Agrobacterium tumefaciens, so that tDNA, we are a Ta plasmid which is get modified into a cloning vector. So no more pathogenic to the plant, but is still able to use the mechanism to deliver the gene of interest into the variety of a plant. So in that Agrobacterium tumefaciens, this Ta plasmid is going to be responsible for the production of a tumor cells. Now we are going to modify this Ta plasmid into a cloning vectors. So we are going to change the infecting property, and then we are going to use only delivering property. So they can able to deliver a gene of interest into a variety of plants. Likewise, retrovirus modified into a disarmed and used to deliver the desirable genes into the animal cells. So either it is going to be a Ta plasmid or a retrovirus. They are going to be inserted with a foreign DNA, and then they can be easily inserted into a plant or a host cell. So one vector for a plant is a Ta plasmid, one vector for animal is a retrovirus. So these two has a common property of delivering property. Now once they become a cloning vector. So before they become a cloning vector, they has a two property. One is a delivering property and then infecting property. So we are going to change that infecting property, and then we are using only a delivering property to deliver the gene of interest to the plants and then animals. So these are the different parts of a cloning vector. So ORI, that is the origin of replication. Here going to be the replication starts. So this selectable markers, they have this antibiotic resistant gene. So we can able to check for the recombinant and then non-recombinant. If it is going to be plated in a antibiotic medium, and then the restriction site is the site which is used to cut the plasmid by help of a restriction enzyme. Then what is the promoter means? The cycle going to be starts during the replication process. This only they are going to start the cycle during the replication process. So coming to the question part, so the figure below is the diagrammatic representation of an E. coli vector, that is a PBR322. So which one of the following options correctly identify its certain components? Option A, HIN3 and then EcoR1, which is going to act as a selectable marker, or ampicillin and then tetracycline resistance, they are going to be act as an antibiotic resistant gene, or ORI, which is an expansion of a original restriction enzyme, or ROP, reduced osmotic pressure. So which one is correctly named according to the B? Because ampicillin and then tetracycline only, they are going to act as a antibiotic resistant gene. So HIN3 and then EcoR1 are restriction enzyme. ORI means origin of replication. So ROP, still now we didn't study about this. So the next question. So which of one of the following bacteria has found extensive use in the genetic engineering work in a plant? Example: Bacillus, Xanthomonas, Clostridium, or Agrobacterium tumefaciens. Agrobacterium tumefaciens only used as a vector for the plants. Then TAE plasmid naturally occurs in which bacteria? Agrobacterium, Corynebacterium, Staphylococcus, or Vibrio? Agrobacterium. The next one, genetically modified DNA fragments are called dash recombinant DNA, sDNA, or mitochondrial DNA, or none of this. Recombinant DNA. So coming to the summary part, so in this schemes we have learned about what are the different parts of a cloning vector and then what is the use of a cloning vectors. So we will be continue the remaining concept in upcoming schemes. Thank you.